Hello and welcome to Learning Target 5.3. In this learning target you're going to be learning about fertilization and development, and you'll be able to explain what happens during fertilization and how an embryo develops into a fetus. Just to start off here, we want to take a minute just to review two different processes that we've learned about so far this year. The first being mitosis and the second being meiosis. And just to draw your attention here up on this diagram, you can see that mitosis is represented here on this side and this side, and meiosis is represented here. Now mitosis is really important because it's what our body does when we grow and when we get hurt or when we need to repair our damaged cells. Meiosis, on the other hand, is really important in order for us to make our sex cells, which we also call gametes. And as a reminder, gametes are the sperm and the egg. And the sperm comes from the testes of the male, and the egg comes from the ovaries of a female. And meiosis only takes place in sexual reproducers. So today we're going to get into this topic here about how mitosis, meiosis lead to the fertilization and development of humans. So all of these different pictures here you'll be able to explain. Now we focus in over here on this side of fertilization. Fertilization is what happens when the sperm and the egg meet. And this happens when the 23 chromosomes of the egg and the 23 chromosomes of the sperm, they join together inside of the fallopian tube of a female or a woman. And when this happens, fertilization occurs and we create a cell which is called a zygote. A zygote is just a fancy scientific name for a fertilized egg. The unique thing about this is that something you have to remember is that an egg and sperm only have half of the chromosomes as an actual body cell or an adult or a human, right? So all the other cells in our body have 46 chromosomes, except for our sex cells, except for our sperm and egg. They have half the number of chromosomes because they were made using meiosis, meaning they have 23 each. But when the sperm and the egg join back together during fertilization, creating a zygote, that 46 chromosomes, or 23 from the egg and 23 from the sperm, are rejoined, and we restore the chromosome number to 46. So just taking a minute to look at this image here, just to make it clear. Sperm and the egg, this is not a human sperm, by the way, you can tell because it has two tails. Human sperm, a normal human sperm only has one. So 23 chromosomes from the sperm, 23 chromosomes from the egg join together, and this process makes what we call a zygote, or a fertilized egg, with the full number of chromosomes, or 46. So sometimes this is represented as an N, and a 2N. So N being 23, 23, 2 times N, or 2 times 23, is 46. And you may see it represented on the regions like this, right? The sperm is actually much smaller than the egg. And when they join together, we get a zygote. Now, one quick thing I just do want to note here about our sex cells and mutations, which is any change in the DNA. Right? Mutations, or when our DNA is changed by something like UV light or the environment or toxins, these mutations or changes in our DNA, they only get passed on to the next generation if they take place in our gametes, or our sex cells. So if it happens in our skin cells or our hair cells, or a mutation in our liver cells, those mutations, they don't get passed on to the next generation because they're not happening in the sex cells or the gametes. Only mutations in our sex cells get passed on to the next generation. Fun fact, right? Now, let's move on to development and implantation, right? If the sperm and the egg meet, right, and this is when the egg gets released from the ovary, it starts traveling down through the fallopian tube. If sperm is deposited in the vagina, it swims up, makes its way through the cervix, into the uterus, and into one of the two fallopian tubes. And if the sperm is lucky enough or strong enough to make it all the way to the waiting egg, they join together, and this is fertilization. And we've created what we call a zygote. Now, once that zygote is created, the process where meiosis, the sperm and the egg were made by meiosis, switches into what we call mitosis, cell division. And as the zygote starts to move down the fallopian tube, it starts to divide by the process of mitosis. And this, remember, creates genetically identical cells. So 46 chromosomes, those go through DNA replication to 92. The cell divides, we have 46 and 46, right? Mitosis happens, those 2 go to 4, those 4 go to 8, 8 go to 16, etc. And eventually, this developing zygote, which is now called an embryo, makes its way into the uterus. And the uterus is where the embryo develops. The embryo attaches to the lining of the uterus, the endometrium, and this is called implantation, and that embryo begins to continue to divide by mitosis and grow into, eventually, a fetus. So just looking at the process here of fertilization and development, the first thing that kicks this process off is when the sperm and the egg join inside of the fallopian tube. So 23 chromosomes from the sperm join the 23 chromosomes of the egg. Those 23 chromosomes now make 46 chromosomes inside the nucleus. We call this a fertilized egg or a zygote. Then mitosis takes over. Mitosis, the cells begin to divide, and eventually those cells begin to specialize or become into the specialized parts that we have, like the liver and the lungs and the heart that we see in our body. Interestingly, all of our cells in our body have the same DNA, but not all of our cells are the same. We have heart cells, liver cells, and lung cells, like I said. The reason we have those things is because different parts of the DNA are used to make those specialized cells. And this process of specialization, let me say that again. This process of specialization has a scientific name that we call differentiation. And inside that word differentiation, you see different, right? Where cells become specialized. So differentiation, like I said, is when cells become specialized based on the different genes and DNA that are used and the different environmental factors that take place within our bodies. So genes, which we're going to learn back in a later unit, can be turned on and off based on where they're located. And when that happens, those genes make different proteins, which then create different types of cells. So we can get smooth muscle cells, or our nerve cells, or our bone tissue, or our striated muscle cells, or cells inside our epithelial lining, or our inside of our digestive system, right? And these cells all have the same DNA, but different parts of the DNA are used because those cells are differentiated or specialized. Now, if you take a look at this Regents question here, you may see something like this on a test, right? This is a very, very popular sequence to show, and we kind of saw it in the previous slide. So let's go through this question together, right? First, anytime I see a picture, I like to try to identify what's going on in each one of these processes. So I would immediately look at this and say, okay, what's going on here is fertilization in reproduction and development, right? So A right here, this must be a zygote. This must be a cell with 46 chromosomes that happen right after fertilization. And then B, we see the cell starting to divide, so this process here is mitosis. And then when we start seeing this kind of thing, especially this folding here, this is differentiation. So if you take a look at the question, right, if this cell is telling us has 46 chromosomes, which we inferred, right, how many chromosomes will be found in each of the cells at stage G? Well, A looks very different than G, but the key thing to remember, right, is that if this cell has 46 chromosomes, then all of these cells, every single one of these cells, are going to have 46 chromosomes. They're all going to have the same exact DNA because this is the process of mitosis. Now, if we look at the second question here, right? The arrow labeled X, the answer here would be then 2. The arrow labeled X, this arrow over here labeled X, right? What does this represent? Well, we already talked about this when we thought about, what is this image showing us, right? This is the process, of differentiation right here. And that's how we make an embryo. So after differentiation occurs, the embryo continues to develop inside of the uterus, right? Sometimes people say, it's in the mommy's tummy, which is not totally true. It's inside their belly, but it's actually inside the reproductive part called the uterus. And then by mitosis, you continue to grow, right? We talked about that earlier in the slideshow, that we have mitosis and meiosis, and mitosis is for growth, which we see happening here when the embryo turns into a fetus, and then eventually develop. You see the little umbilical cord poking out here, which attaches to the placenta, where the nutrients are exchanged. And this is actually what a placenta looks like. I've had the experience of seeing this up close and personally 3 times for my 3 children. This is actually a nice, really interesting color, but my kids didn't actually look like that. But anyways, that's a story for another time. But the important thing you need to know about the placenta is that this placenta is inside the uterus. You can kind of see it over here, right? And the thing that's interesting about the uterus is that this is where the nutrients and oxygen and waste get exchanged between the developing fetus and the mother. Now, the thing that you need to remember is that the blood of mom and baby never mix, and that's because we have a placenta. But the blood of mom and baby become so close together that the nutrients and the waste and oxygen and carbon dioxide, they can be exchanged by the process of diffusion when molecules move from a high concentration to a low concentration. And this again is happening inside the uterus. There's always going to be a question on your test about the placenta, so it's really important to know about this process and what happens with the placenta. The last thing, maybe one of the last things to note here, and this is pretty common sense, but just as a public service announcement, I'll mention it, right? If a mother is ingesting or around toxins or alcohol while she is pregnant, those things can impact the growth and development of the developing fetus. And the way this happens is by the same thing that waste and nutrients and things like that are exchanged. A lot of these things and substances like toxins and alcohol are inside of a mom's blood. And they diffuse across the placenta and ultimately end up inside of the baby. Now, these substances are much more dangerous to a developing baby because the baby is so much smaller than the mom. So while one alcoholic beverage may not impact the health of a woman all that much, the effect on a much smaller baby could potentially be much more significant. So that's why it's very important, especially if you're pregnant, to avoid toxic substances because it impacts the development of the fetus. That brings us to the end of Learning Target 5.3. Thank you for listening.